Hi everyone, welcome to lesson 3.8 on the M phase. So we're going to start with a little bit of a review. This was in the last lesson, so I'm going to go through this really quickly. But if you remember, last class we talked about chromosomes, and remember chromosomes are thread-like structures of DNA and protein that contain genetic information. In prokaryotes, here's a good diagram of typically what a prokaryotic chromosome looks like. So remember that consisted of a single circular chromosome, and it contains all the cell's genetic information, and it's found in the cytoplasm. Eukaryotes, we know, are more complex, and the chromosomes are usually found in the nucleus. So here was all that information from last time on chromosomes, how they're packaged. Okay, if you don't remember that information, go ahead and read through this or go back and re-watch that from last lesson. But here's a really good diagram, and we'll just kind of go back through the structure of a chromosome. So this all together, okay, is the chromosome. All right, chromatin was all that condensed genetic information, again, kind of like spaghetti, and condensed chromatin is what makes up the entire chromosome. The chromosome, okay, has two here. This is a chromatid, and this is a chromatid. So kind of think of them like the two legs. So some chromosomes have what we call sister chromatids, and in this case, this is a sister chromatid, and this is a sister chromatid. These sister chromatids are connected in the middle by a centromere. Okay, so that kind of holds the sister chromatids together. And the last part of chromosomes that we talked about were the telomeres, okay, which are the ends of eukaryotic chromosomes. And again, you can kind of think of those like the ends of shoelaces. So now we've seen this quite a few times. But if you remember the eukaryotic cell cycle, we have two main phases, interphase and the M phase. And today our focus is going to be on the M phase. So interphase, the big thing is last lesson, we went through DNA replication. Okay, so we went through that S phase in more detail. And remember, interphase is a time of growth. DNA replication checks before the cell goes into that mitosis or into that cell division. Again, making sure that there's any repairs and errors, so that way that cell isn't duplicated if it's damaged or something. All right, so remember, DNA replication, growth, interphase is extremely important and has to be done correctly in order for us to enter the M phase. So that's going to be our main focus of today. So the M phase consists of two parts. We have mitosis and cytokinesis. Now, mitosis is the cell division, okay, during which the nucleus divides. Cytokinesis is the division of the cytoplasm to form two separate daughter cells. Now you do need to know the 4 stages of mitosis. And here's going to be how we remember those. Okay, so say it with me. PMAT. All right. Say it one more time. PMAT. So the 4 stages of mitosis are prophase, metaphase, anaphase, and telophase. And we're going to look at each of these stages in more detail. So first up, we have prophase. Prophase is the first and longest phase of mitosis in which the genetic material inside the nucleus condenses and the chromosomes become visible. So down here is more details. Okay, you don't have to know every single one of these details. This just kind of helps us identify prophase a little bit better. The big thing is making sure that you have the definition, and if given a diagram or something like this, you should be able to identify its prophase. Okay, so that's why in here we made the diagrams much larger because you might have to identify those. So the duplicated strands of the DNA molecule can be seen and are attached along their length at these centromeres. So remember, that's like the middle, okay, of those chromosomes connecting those chromatids. And it says right here, recall each DNA strand in the duplicated chromosome is called a chromatid. The cell starts to build a spindle, okay, which is a fan-like system of microtubules that help separate the duplicated chromosomes. Spindle fibers extend from a region called the centrosome, okay, and where tiny paired structures called these centrioles are located. Plant cells lack centrioles and organize spindles directly from their centrosome regions. Again, a little bit more detail than what you need to know, so don't worry about those. As prophase ends, the chromosomes coil more tightly, the nucleolus appears, and the nuclear envelope breaks down. So that's kind of what you see here with these dotted lines. That's showing the nuclear envelope starting to break down. Next up, we have metaphase, and that is the phase of mitosis in which the chromosomes line up across the center of the cell. So the spindle fibers connect the centromere of each chromosome to the two poles of the spindle, okay? And whenever you see metaphase, think middle, right? So metaphase, think middle. They line up here in the middle of the cell, okay? And it says these spindle fibers connect the centromere of each chromosome to the two poles of the spindle. So metaphase, line up in the middle of the cell. Next up, we have anaphase, and that's the phase of mitosis in which chromosomes separate and move to opposite ends of the cell. So anaphase begins when the sister chromatids separate and begin to move apart here. Once anaphase begins, each of the sister chromatids is now considered an individual chromosome. So even though they're now separated and not that X shape that they were, this is still considered a chromosome. Okay, anaphase has ended when the chromosomes are completely separated into two groups. So whenever you see anaphase, think apart, all right? The chromatids moving apart, becoming their own individual chromosomes. Last phase of mitosis is telophase, and that's the phase of mitosis in which the distinct individual chromosomes begin to spread out into a tangle of chromatin. So a nuclear envelope reforms around each cluster of the chromosomes. The spindle begins to break apart, the nucleolus becomes visible, and each daughter nucleus. And after this stage, mitosis is complete. But remember, the M phase consists of both mitosis and cytokinesis, so we have one more step. So cytokinesis, again, is the final phase of the M phase, and it's the division of the cytoplasm to form two separate daughter cells. So remember, mitosis produces two nuclei, each with a duplicate set of chromosomes, and then cytokinesis is what divides the cytoplasm. So the process of cytokinesis differs in animal and plant cells, just a little bit depending, because again, plant cells have that cell wall, so they perform cytokinesis a little bit differently. In animals, we have something called a cleavage furrow that kind of pinches the parent cells into two. And in plants, we have a cell plate. Again, because we have that cell wall, it's a little bit harder to divide, so the cell plate instead does that for us. So here, go ahead, pause, and try to identify the stages of the cell cycle. So this diagram does include both interphase and cytokinesis. So try that out. Okay, so if you're looking at this and you're like, I don't really know where to start, that's fine. Okay, look at something to where you can recognize what it is. So here, obviously, we see that those are lined up in the middle. So this would be metaphase. Okay, what comes before metaphase? Prophase. Okay, so prophase. What comes before prophase? This would be interphase, and we can tell that's interphase because if you look kind of closely in here, the chromosomes haven't condensed yet, right? They're still in chromatin, okay? They don't condense until we come to prophase. So prophase, metaphase, lined up in the middle. Anaphase, these things are pulling apart. Telophase, okay, nuclear envelopes starting to reform. And the last one, cytokinesis, okay? And there, that would be that cytoplasm splitting, okay, back to interphase. So if given something like a diagram, you should be able to identify what stage of mitosis or cell cycle that we are in. So this was also shown last class. Again, this is just kind of a good overview about the process of DNA replication, and then after DNA replication, kind of what happens. So DNA replication, remember, occurs during interphase before mitosis even happens, but the two copies of DNA produced in replication during the S phase remain closely associated until the cell enters prophase of mitosis. Again, once it enters prophase, the chromosomes condense, and the two chromatids in each chromosome become visible. They separate from each other in anaphase of mitosis, and that's producing two cells, each with a complete set of genes coded in DNA. So replication, again, kind of gets us ready to perform mitosis, so that way after cytokinesis occurs, they each have a complete set of genes and DNA. Okay, so here's our exit ticket. Go ahead, pause here, and try these on your own. All right, so stages of mitosis. Always think PMAT, prophase, metaphase, anaphase, telophase. The division of the cell nucleus was mitosis. The division of the cytoplasm was cytokinesis. And DNA was replicated during the S phase during interphase. The difference between animal and plant cells during cytokinesis. Remember, animals form a cleavage furrow, and plants just form a cell plate. So that is the end of lesson 3.8. Thanks for watching. Reach out with any questions.